Hello, my name is Liana Tang. This fall I will be attending South Point Catholic High School as a sophomore. So this summer I worked in Dr. Lichty's lab alongside Fariba Hossein, who's part of the MSRP program. And I was researching wound healing and regenerative medicine in diabetic wounds through the manipulation of microRNAs.
Good morning, everyone. My name is Esmeralda. I am currently a junior attending Padilla High School. This summer, I worked in Dr. Lichty's lab alongside Jimena Ganchis and Marcy Ojeda. In the duration of the summer, we worked on culturing and reproducing cells from both wild-type and diabetic mice from harvested skin tissues.
Hello, my name is Natalia Padilla. I'm an incoming senior at UC Berkeley, and this summer I also worked in Dr. Lichty's lab with Dr. Anisha Abdi, and we looked at injured versus healthy sheep uterine tissue after C-section.
So my beginning research question was how the manipulation of anti-angiogenic factors such as microRNA 15B impact the diabetic wound impairment.
My initial question is, what exactly is the physiological process in which diabetic cells decrease the fibroblast production in organisms?
My beginning question is, what are the molecular mechanisms responsible for the mechanical characteristics of sheep uterine tissue? So for the first step of RNA extraction, I'm going to be taking 1 ml of Quizal and adding it to our samples.
So now that I have my sample, we're going to homogenize it for 15-second intervals on low, medium, and high speed.
So the next step for RNA extraction is to add 200 microliters of chloroform, and then after that we will then vortex it.
So now we're centrifuging our samples for 10 minutes, and they should be balanced.
Right now we are trying to remove the supernatant, which is the top layer of the sample, and then transfer it into another tube.
The next step is to add isopropyl, which should be 60% of the supernatant value.
Now we're going to centrifuge our samples.
So day 2 of the qPCR protocol is pellet suspension. So I'm going to start off by removing the supernatant and putting it in the waste bottle.
And now then nanodrop our samples.
Now I'm aliquoting 10 microliters of our sample into separate tubes for the dilution.
We then did the calculations to determine how much water we will then add into the sample to dilute it.
After diluting our samples, we're going to re-nanodrop our samples to see the RNA concentration.
So now I'm going to add DNase buffer into this tube to create the DNA master mix. So now I'm going to add the DNase enzyme into the master mix.